Welcome everyone, and we're going to continue our discussion on bioenergetics. In part 4 of this series on chapter 3, we're going to discuss ATP production, more importantly, the aerobic pathway where carbohydrates are the main source of fuel. But before we begin, let's go ahead and do a quick review. Over the past few videos, we've discussed about how the body has stored ATP. It can create ATP through the phosphocreatine pathway, and then we can also create ATP via glycolysis via the absence of O2 or oxygen. In this particular video, we're going to talk about how ATP is produced via the process of oxidative phosphorylation. And what this means is ATP is produced in the presence of oxygen. This is the key difference between aerobic versus anaerobic processes. Now, before we get into it, I want to discuss a couple of key concepts that we need to know about oxidative phosphorylation. First off, this process can utilize carbohydrates or non-carbohydrate sources as the fuel source. But in this particular lecture, we're going to focus primarily on the fuel source of carbohydrates as the fuel source for ATP synthesis through oxidative phosphorylation. We'll talk about the other fuel sources in the next video. The next concept to understand is that oxidative phosphorylation uses glycolysis in the initial steps of ATP synthesis. And we'll cover that and the key differences between this and the anaerobic glycolysis. And finally, in the previous video, we discussed the concept of electron carriers. And this is where they become very important. And you'll come across two key electron carriers. The first one is called nicotinamide adenine dinucleotide, or NAD+ for short. The second one is also named flavin adenine dinucleotide, or FAD+. And we'll talk those differences in their roles with oxidative phosphorylation as we review the process. So, in discussing glycolysis, there's two little concepts you need to remember. The first is glycolysis always uses carbohydrate as its fuel source. And this is depicted as glucose as the entry point into our glycolytic pathway. The second point is the rate-limiting enzymes that help modulate the glycolytic pathway. And we'll discuss those as we review the glycolytic pathway. So let's go ahead and review the glycolytic pathway. In the previous video, we stated that the first half of the glycolytic pathway involves energy investment. And this is where we invest 2 ATP in order to create further ATP in the absence of oxygen. And so with this, within the first steps, glucose is converted into the molecule G6P via the enzyme of hexokinase. Then glucose 6-phosphate is converted into fructose 6-phosphate. And then via the enzyme phosphofructokinase, or PFK for short, we utilize another ATP and then create fructose 1,6-bisphosphate. Now, from there, the fructose 1,6-bisphosphate is split into 2 different molecules. And this is done via the enzyme adulase. And this ends the energy investment steps and gets us into what we call the energy harvesting steps. And during the initial steps of the second half of glycolysis, adulase converts the fructose 1,6-bisphosphate into the molecule glyceraldehyde 3-phosphate, or G3P for short. And then from there, G3P is converted into 1,3-bisphosphoglycerate, or BPG. And at this point, we start seeing our first interactions of the electron carriers. And what we have is we have two molecules of NAD being oxidized to NADH. And these will carry off the electrons over to the electron transport chain to create ATP. And we'll discuss that later on in this video. And then moving forward, then when 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate, we create two molecules of ATP. From there, 3-PG is converted into phosphoenolpyruvate, or PEP for short. Then PEP is converted into pyruvate via the enzyme pyruvate kinase. And pyruvate kinase creates two more ATP via this process. So to quickly review the products that are brought forth through the glycolytic process is, first, we actually created 4 ATP, but we used 2 ATP in the first half of this process. So we have a net gain of 2 ATP through the glycolytic process. Furthermore, we also created 2 NADH, which will in turn help create 5 ATP via the electron transport chain. So far in our review of glycolysis, there's been no difference between the glycolytic pathway with the absence of oxygen or in the presence of oxygen. This becomes important at the step of the creation of pyruvate. At this point, we have two different things that can occur. So without the presence of oxygen, pyruvate is converted into lactate plus acid, or a hydrogen ion, via lactate dehydrogenase, or LDH. Now, in the presence of oxygen, pyruvate is converted into the molecule acetyl-CoA. And by this conversion into acetyl-CoA, the two molecules of acetyl-CoA can now be transported into the mitochondria. Because remember, glycolysis occurs in the cytoplasm of the cell. And so as we're moving into the mitochondria, one thing that occurs right before that is, as pyruvate is converted into acetyl-CoA, we create another molecule of NADH. And keep this in mind when we're talking about our total tally of ATPs produced during the entire oxidative phosphorylation pathway. Also, this is the point where non-carbohydrate sources such as fatty acids and amino acids can enter into the oxidative phosphorylation pathway. And we'll discuss how that occurs in a subsequent video. And so when acetyl-CoA enters the mitochondria, it does so from the cytoplasm into the matrix of the mitochondria. And so this blue area here on this diagram is the matrix of the mitochondria. And this is where the CAC, or the citric acid cycle, also known as the Krebs cycle, occurs. And so as acetyl-CoA enters in the citric acid cycle, it is converted into the molecule citrate. From there, it's converted into the molecule of isocitrate, then into the molecule of alpha-ketoglutarate. And at this point is where we create our first NADH of the Krebs cycle. From there, alpha-ketoglutarate is converted into succinyl-CoA. And again, we create our second NADH. Then, succinyl-CoA is converted into succinate. At this point, we create our one molecule of ATP through the citric acid cycle. From there, our succinate is then converted into the molecule of fumarate. And during this conversion, we get our production of our 1-FADH. From there, fumarate is converted into malate, and then malate is converted into oxaloacetate. And during this process of conversion of malate to oxaloacetate, we create our third NADH. So to sum up our products during this entire process, we create 3 NADH, 1 FADH, and 1 ATP. And so if we do our math using our conversion factors, so for example, we know NADH creates 2.5 ATP per 1 molecule of NADH. Additionally, we know FADH on average creates 1.5 ATP per 1 molecule of FADH. Also, we need to keep in mind that we have 2 molecules of acetyl-CoA that go through the Krebs cycle. And so what that means is we need to multiply the FADH and NADH quantities by 2. And so this gives us a total of 6 NADH created through the process and 2 FADH created through the process. And so if we multiply NADH by our factor of 2.5, this will give us 15 ATP produced throughout the Krebs cycle. And then if we multiply the FADH by our factor of 1.5, this will give us 3 ATP created through the Krebs cycle. And if we sum these up together, the electron carriers give us a total of 18 ATP produced through the Krebs cycle. And then if we add in the 2 ATP that are created by the 2 acetyl-CoA that go through the Krebs cycle, we add those in, we get a total of 20 ATP created through the Krebs cycle. And so as we're moving away from the Krebs cycle, the electron transporters move from the matrix of the mitochondria into the inner membrane. And this is where the electron transport chain is located. And so as we look a little bit closer at the electron transport chain, it is made up of 4 different complexes. And they're simply known as complex 1, complex 2, 3, and complex 4. And sometimes complexes 1, 2, and 3 are known as pump 1, pump 2, and pump 3. Either way, the names are interchangeable. And so what happens is the electron carriers of NADH and FADH bring their electrons to the pumps to create ATP. And so what happens is as the hydrogen ions are reduced from NADH, they are pumped from the matrix into the inner membrane. And what this does through this entire process, through the different complexes, is create an electron gradient. And so what is happening is as these hydrogen ions are pumped from the matrix into the inner membrane space, the inner membrane space is becoming more and more positive than the matrix itself. And so this creates an electron gradient. And as we reviewed previously, with gradients, you want to go from areas of high to areas of low. And that's what we're going to see here in complex 4, is these electrons are going to want to go from the gradient of high positivity to the matrix. And what this does is they're passed through pump number 4, and this provides the energy for ATP synthase to create ATP. And this is the whole reason behind of the electron carriers is because they provide the energy necessary to create ATP. And so if we look at complex 4 a little bit closer, we can get a better view of the ATP synthase. And so this whole enzyme right here that I'm highlighting in red is ATP synthase. So here on the bottom part of the figure, we have the inner membrane space, and here we have the matrix. And as we stated earlier, in the inner membrane space, we have a higher positive electrical gradient in the membrane space versus the matrix. And so what this does is creates an electrical gradient, forcing the hydrogen ions to want to go from the inner membrane space to the matrix. And so what happens is the hydrogen ions pass through the ATP synthase, powering the enzyme. And as they pass through, giving the enzyme power, it rotates. And so in each of these pockets of the ATP synthase, you'll have an ADP right in here, and then you'll have a phosphate in the next one. And so as it rotates together, it creates ATP. So at the end of the oxidative phosphorylation process, with one glucose molecule, we can create a total of 32 ATP. And so how do we get to this? Let's break it down into our steps. First off, during the glycolytic process, we create a net of 2 ATP. Remember, we invest 2 ATP but create 4, so that gives us a balance of a positive 2. Then we also create 2 NADH. And if we multiply that by a factor of 2.5 ATP, we get a total of 5 ATP produced through the glycolytic process. Then as pyruvate is converted into acetyl-CoA via pyruvate kinase, we get 2 more NADH. And as we multiply that by our factor of 2.5, we create 5 more ATP. And then during the Krebs cycle, we discussed you can create 2 ATP directly and kind of glanced over this molecule GTP. And the reason we do this is the reaction is instantaneous. So this gives us 2 ATP directly. And then further on, we stated that you can create 6 NADH and 2 FADH. And we went through that math. And so if you put these all together, this is how you get 32 ATP. So we've covered a lot in this video. And so let's try to summarize what we've covered and some of the key takeaways that you need to remember in order to be successful throughout this course. First off, know the difference between aerobic glycolysis and anaerobic glycolysis. And what we mean by this is in the presence of oxygen, pyruvate is converted into acetyl-CoA. And without the presence of oxygen, it's converted into lactate and hydrogen. Secondly, know the glycolytic rate-limiting enzymes. And again, these are hexokinase, PFK, or phosphofructokinase. And the third one is pyruvate kinase. We do talk about the enzyme of atolase, but that is not a rate-limiting enzyme. Thirdly, you need to know the molecules in which glucose is converted into during glycolysis and the citric acid cycle. And so what this means is you need to know the name. So what this means is you need to know that glucose is turned into glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, G3P, BPG, 3PG, PEP, and pyruvate. And this is just for the glycolytic cycle. And then during going into the Krebs cycle, you need to know that pyruvate is converted into acetyl-CoA, and then you need to know the names of the subsequent substrates, such as citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. Additionally, you need to know where the electron carriers are created. For example, right here where this NADH is created when pyruvate is converted into acetyl-CoA. Also, where they're created during the Krebs cycle, and also during the glycolytic cycle. Next, you need to understand how the electron transport chain works. And by this we mean about the electron gradient, how the hydrogen ions pass through the different complexes, and how ATP synthase works. And finally, you need to know how many ATP are created during the oxidative phosphorylation pathway and be able to perform the math. And what is meant by this is know how to calculate how many ATP are created by the electron carriers of NADH and FADH, and where the ATP is directly created from, and how to get to this number of 32 ATP. I hope this helps. Please let me know if you have any questions. Take care.